So, good morning, students. In the previous lecture, we had a discussion about different models of replication. And if I have to revise briefly, I would like to say that there are three different models of replication which have been, which were proposed earlier after the discovery of double helical structure of DNA by Watson and Crick. And the three different models are, one is called as conservative model, another one is called a dispersive model, and third one is called as semi-conservative. We have discussed all the three models in the previous lecture. So, and the conclusion of the previous lecture is that DNA replication is semi-conservative in nature, which was confirmed by Michelson and Stahl in their experiment. So if I have to briefly explain their experiment, I would like to show you the diagram. This is the diagram which gives the brief description of Michelson and Stahl in 1957. They carried out their experiment to demonstrate the nature of the DNA replication. And it is now very clear that DNA replication is semi-conservative in nature. What they did is they carried out the DNA extraction on bacteria by name E. coli. And what they did is they initially cultured the E. coli in a medium containing nitrogen 15 as a source of nitrogen. Then the culture was taken again and was grown on the medium which contained nitrogen 14. Similarly, it was carried to the next culture and next culture. And DNA from each culture, in the very first culture, DNA showed the presence of the nitrogen 15. As you know, the nitrogen's base, if you go to the nucleotide structure has three components. One is pentose sugar, phosphate, and the nitrogenous base, the adenine, guanine, cytosine, and thymine. And those bases are rich in the nitrogen, and that nitrogen comes from the source. Okay. If the source of nitrogen is 15, by default, nitrogen 15 will be present in the DNA. And they found that this DNA contains nitrogen 15, yes. And when these E. coli containing nitrogen 15 were cultured on the nitrogen 14 medium, it was found that in this one, that the DNA contained both nitrogen 15 and nitrogen 14. And further culturing, the DNA isolation led to the discovery that now the DNA contains more and more 14 and the content of the nitrogen 15 is diminishing. Because why the nitrogen 15 is decreasing and diminishing? Because now the bacteria are continuously being cultured in a medium which contains nitrogen 14, not the nitrogen 15. Okay? Let me tell you, nitrogen 15 is a heavier isotope than nitrogen 14. And if you look at the last one, we have three DNA strands which contain nitrogen 14. And the last one is, it has 50-50. One strand is nitrogen 14 and another one nitrogen 15. So it was demonstrated that successfully that DNA replication is semi-conservative in nature. And if you look at this double helical strand, one strand comes from the old strand. This 15 has come from here, from the first culture. That's why that's what we call as 50% of this DNA. It has two strands. One strand belongs to the old one, and the one strand is a newly synthesized strand. So we call it semi-conservative. Half of the DNA is conserved here. In the double-stranded DNA, one strand belongs to the old one, and the one strand belongs to the newly synthesized one. I hope this is clear to you. The few things which you must memorize, that when questions are asked in such type of from such type of topics, the first question which is usually asked, which are the following organisms were used for Michelson and Stahl? It's E. coli. Which are the following nitrogen isotopes were used? Nitrogen 15 and 14. These two questions you must remember. Okay, and let me tell you, DNA was centrifuged. Gradient centrifugation was done in cesium chloride solution, CSCl. The centrifugation of the DNA gradient centrifugation. Because you see, if for example, if I tell you, if this is a test tube, if this is a test tube, if you centrifuge the test tube, there are two elements present in the test tube. If you do the gradient centrifugation, the one which is heavier will settle at the bottom, and the one which is lighter will settle above this. Okay, nitrogen 15 being heavier will settled at the bottom, and nitrogen 14 being lighter settled at the top, settled above the 15. So they carried out the nitrogen gradient centrifugation after the extraction of the DNA. In cesium chloride, remember that gradient centrifugation in cesium chloride was carried for each culture, okay, to segregate the nitrogen 14 and nitrogen 15. To understand the mechanism of DNA replication, whether it's conservative or semi-conservative. I hope this is clear. Now, we have these were the models of DNA replication, okay? Now we have modes of DNA replication. There are different modes of DNA replication. For example, see, if you look at bacteria, you have a circular DNA. DNA replication is, the mechanism of replication is different. The mode of replication is different. If you look at human beings, you have the kind of this one. You have a kind of linear chromosome, right? So you have a different kind of replication. Based on the structure of the genetic material, we have different modes of DNA replication. Okay. Now, one more thing you have to remember is, for example, as I said in the previous lecture, template. You see, if this is a double-stranded DNA, this is a double-stranded DNA, okay? Let me see the double-stranded DNA. This is a double-stranded DNA, okay? And when this undergoes replication, it has to unwind. This is tightly coiled. The bases of one strand are attached to another strand. Fine. Before replication, this is tightly coiled. It has to unwind, right? And these unwound strains, single strand is called as template. This serves as a template. This will give rise to new. With the help of this, we will synthesize new DNA strand. We will synthesize new DNA strand with the help of this. This is called as template. This is called as template. See, we have studied different modes of replication. See, we have different kinds of DNAs. For example, if we look at bacteria, we have circular DNA, double-stranded circular DNA. We have in virus and other small prokaryotes, we have some single-stranded DNA. And in eukaryotes like you and me, we have a linear DNA in the form of chromosomes. Okay? Yes. So based on the different kinds of structure of the DNA present in the nucleus, we have different modes of DNA replication. Okay. And for example, see, this is a DNA replication. This is a DNA, circular DNA. Tightly count circular DNA. So if this DNA undergoes replication, this circular DNA has to unwind first. You go like this, you go like this. Something will happen like the circular DNA. Okay. And this circular DNA, one circular DNA will become what? Template. Here the template is circular DNA, right? Here the template is circular DNA. In human beings, for example, if you have this, when they will unwind the template, here you have linear DNA, right? Depending upon the template, you have different number of replication forks. What is the replication fork? Before that, you have to understand origin of replication. For example, I will give you the example of a human eukaryotic chromosome. Please understand it. This is your chromosome. Okay? This is your DNA. It has to undergo replication. From which point the replication start? That's a big question. That is a very big question. From which point the replication start? Whether it will start from here, whether it will start from here, or whether it will start from here, whether it will start from wherever it starts, that is called as origin of replication. If it starts from here, from this point it will start. This point is called as origin of replication. This point is called as origin of replication. This point is called as origin of replication. Origin of replication is a point where the replication starts. Where the replication starts. Am I clear? For example, if I talk about this is circular DNA, right? This is circular DNA. This is very much circular DNA, and from here the replication starts. I will say this point is called as origin of replication. This is called as origin of replication. So origin of replication, ORIC, origin of replication is a point where the replication starts. Now the question is, sir, how can we say the origin of replication is here? That all we have to discuss in the mechanism of DNA replication. We are just trying to understand different modes of replication. In bacteria, we have a different mode. In virus, we have a different mode. And in human beings, we have a different mode of replication. So one thing you have to understand is that first thing, the mode of replication principally depends upon the nature of the template. If the template is circular, we will have a different kind of mode of replication. If the template is linear, we will have a different mode of replication. And in each organism, whether it's a prokaryote or the eukaryote, we have different number of origin of replications. We have different number of origin of replication. Remember one thing, for prokaryotes, bacterial chromosomes, we have only one origin of replication. For example, if this is your double-stranded DNA, if this is your double-stranded DNA in bacteria, there's only one origin of replication, only one origin of replication. However, in human beings, see, we have a huge genetic material. You might have seen in the chromosomes. Our nucleus contains a huge genetic material, very complicated. Chromosomes is a very complex structure. And see, this is bacteria. Only one ORIC, only one replication origin is there. That means only this point replication starts. Only one. However, if you talk about human, for example, if this is a chromosome, you have many origin of replications in human beings. See, we have a large chromosome, we have large number of, we have a large genetic material, huge genetic material. So in order to complete the replication in due time, replication starts at many points. So we say in a linear chromosome, in eukaryotes, there are many origin of replications, and in prokaryotes, only one origin of replication is there. See, tell you. Now remember, this thing is to be memorized. In prokaryotes, you have only one origin of replication, and in eukaryotes, you have many origin of replication. The reason being, eukaryotes have a large genome, very large genome. Quantity of the genome is very high. However, in prokaryotes, you have a very small genome. So you don't need much time to replicate. As you know, in a fraction of seconds, the bacterial cell divides. Okay. So please understand this. Origin of replication is the point where the replication starts. And number of replication origins is different in prokaryotes than in eukaryotes. Is that clear? Please, you may please unmute the microphone for a while. Is it clear?
Yes, sir. Yes, sir.
So origin of replication is the point where the replication starts. We have different modes of replication based on the template. See, in prokaryotes, the DNA is circular. That means the template will be circular. In eukaryotes like you and me, the DNA is linear. We have the DNA in the form of chromosomes. So we will have a linear template. So modes of the replication will be different. So first mode of replication we call as theta replication. That's called as theta replication. And let me tell you, theta replication what is this called? This is theta, no? We call this theta. This is theta, no? This is theta. I will tell you why this is called as theta replication. Before that, I want to tell you that theta replication takes place in prokaryotes. It takes place in E. coli, bacteria. Remember that. Theta replication, the mode of theta replication is found in bacteria. It's found because here the DNA is circular in nature, unlike you. What happens in this? You have a circular DNA. Yes. It starts unwinding. And once the two complete replication is complete, you have at the end, you have like this replication. This is the this you call as these are two newly synthesized. So and it resembles theta. That's why it's called as theta replication. How all this happens, we have to discuss it. Now what happens? I will explain you in the diagram. But before that, please remember that theta replication takes place in E. coli. That means in bacteria. Okay. Why is it called as theta? Because if you once we go to the next slide, when we will try to understand the structure, you will see at the end of the replication, the structure looks like theta. Looks like a word theta. That is the reason it's called as theta replication. So what happens in this replication? Okay. So we have double-stranded DNA, circular DNA. It starts to unwind because see the DNA is circular and the DNA is supercoiled, right? And I have been telling you guys that when the DNA has to undergo replication, it has to unwind first. So when it starts unwinding at origin of replication, it produces a single nucleotide strand, right? Which serves as the template on the DNA can be synthesized. We have a double-stranded DNA. Yes. This is a double-stranded DNA. So before it starts synthesizing, it starts replication, it has to unwind. It has to unwind from somewhere. It has to unwind. When it unwinds, so you will get two different nucleotide strands, right? So this will serve as a template. This will also serve as a template, but these are circular in nature. This will synthesize one strand. This will synthesize one strand. Both the nucleotide strands will serve as template, and both will serve as template, and both will synthesize a new strand. Okay, because each strand serves as a template, and a template can synthesize a new strand. Okay, and once both the strands start unwinding, and they form a loop that called as replication bubble. The origin of replication, at the point of origin of replication, a bubble-like structure forms that you will understand in the diagram. And when the bubble-like structure forms, unwinding proceeds, and the bubble becomes larger and larger. And as you have two strands, you have two nucleotide strands. DNA takes place on this also and on this also. DNA takes place simultaneously on both the strands. Okay? And when both the nucleotides meet, see, synthesis on the upper strand goes from here, and from the lower, it goes from here. I said 3 end and 5 end are opposite in direction. I have told you in the structure of nucleotides. And this 5 to 3 will go here, okay? And this will go 5 to 3 here, 3 to 5 here. When both they will meet and they will form fork-like structure, it will be like this, and this becomes theta. This is difficult to understand for you guys. So before you get bored, I will directly go to the diagram, theta replication. And let me tell you, it was John Cairns who first provided the visible evidence of theta replication by growing the bacteria in a radioactive nucleotides. Remember that. Okay, John Cairns. He grew bacteria in radioactive nucleotides where he could continuously monitor the different nucleotide strands. Okay. Now coming to the theta. Yes. Now look here, please. Look at this. Very carefully. This is your double stranded DNA, right? This is double-stranded DNA. Understand this. This is the origin of replication. I said from here the replication will start. So when I zoom in on this, it is like this. It is a double helical structure. So it has to unwind. It has to unwind. Now coming to this, it has to unwind because origin of replication is here. When it unwinds, it will give rise to bubble. See, this is the bubble. This it has to unwind. This bubble I was talking about, right? And this is called as replication bubble. The gray is your old DNA. The gray, see, this is a double-stranded DNA. This is double-stranded DNA. I said when these double-stranded DNAs are separated, each strand becomes a template, and each template strand will synthesize a new DNA. See, now this gray strand is separate. These are the old strands. This is a template. This is also template. And this template will synthesize new strand. This template is the new strand. Both the templates are synthesizing the strands. And now it's a complete, and it becomes theta here. And now you have the DNA, complete newly synthesized DNA. See, the gray is the old one, and this is a newly synthesized strand. This is a gray, and this is a newly synthesized strand. You have two. This is here. Look at the structure here. It looks like theta, right? It is a theta-like structure. That's why it's called as theta replication. I will explain this again. How? This is your DNA. This is your circular DNA, right? I said in prokaryotes, we have one origin of replication. Origin of replication is a point where the replication starts. This is the origin of replication. And if you go inside the origin of replication, inside the DNA looks like this. This is a oiled DNA inside, double helical DNA. So as I have been telling you earlier, in order to set out replication, this has to unwind. This has to unwind, right? This has to see. This is wind. This has to unwind. And when it unwinds, the two strands will separate. The bubble will like structure. And when the two nucleotide strands will separate, they will become individual strands. The individual strand becomes a template. A template is what? A single DNA strand which synthesizes a new DNA, a new strand, which synthesizes a new DNA strand. That is a unique property that we will understand in the coming lectures. This one, because you see, complementary base pairing. If this is a DNA template, it is A here, adenine is here, thymine is here, cytosine is here, guanine is here, okay? And other bases will come, they will make a DNA. G, this is A, because these bases have a tendency to form bonds with each other, complementary base pairing. We had a discussion about this in the lectures probably in the last week. So this will uncoil, replication bubble will form, the two strands will separate, and each strand will serve as a template. You see, the bubble will deepen. See, the bubble will go deep and deep, and the new DNA will be synthesized. The maroon is the new DNA, and the gray is the old DNA. It's the template DNA. Okay. Now complete synthesis. Here the structure looks like theta. Let's call it theta replication. So, and theta replication is a type of replication common in E. coli and other organisms possessing circular DNA. Is this clear to you with this diagram? You can have a look on the diagram again. Bubble replication, origin of replication, replication bubble. The two DNA strands serve as a template, and each template synthesizes a new DNA strand. And this maroon is the one synthesized by the, let's see. Right? Now, one more thing I just want to tell you. If this is 5 here, 5 end, this is 3 end here. You just recall the lecture when I was trying to explain the linear structure of DNA. if this DNA is 5 to 3, and then this will be opposite. This will be 5 from here and 3 from here, right? Do you remember that? I said, and you know that the DNA synthesis occurs at 3 position. So this will go from here, and this will go on synthesizing from here. I'm just telling you the example. Okay. So both will be synthesizing in opposite direction. As you know, at the pentose sugar 3 carbon, the nucleotides get added up. That I was telling you that time, you have to use this 5' to 3' concept. And now one thing should be clear, the DNA synthesis is always in 5 to 3 direction. 5 to 3 direction. 5 to 3 direction, right? Simultaneously on both the strands, the DNA is synthesizing. Is that clear to you? Please. Any doubt? This theta replication.
Yes, sir.
Girls,
No, sir.
you look at very minutely, you look at this. Huh? I will share my video also. Do you visit Google Classroom or no? Do you visit Google Classroom? Yes or no? Because every day I share my video on the Google Classroom, and I expect you guys to go through that and also be ready for the, what is that called as presentation. So this is theta replication. This is the diagrammatic representation of theta replication. Now coming to one more application, we have one more application that is called as rolling cycle replication. Okay. Again, I think it is better to go to the diagram and explain your rolling cycle replication. It is found in virus and some other cellular organelles. See, this is your double-stranded DNA. What happens here? A cut is made on one of the two strands. See, there are two strands of the double strand is there. On the above strand, the cut is made, and it exposes your three end and 5 end. And you know the replication, the synthesis of DNA will be at 3 and not at the 5 end. See, this goes like this, again, and this DNA 5 part, it is released as a free in the nucleus. And once it is released as free, it becomes a template. It synthesizes its own DNA inside. This also synthesizes its own DNA inside. Here, the cycle may be repeated if it like this, but you have the two. Why it is called as rolling cycle? I will tell you. You see, you have a double strand, yes. Here the above strand is broken. A break is given. A cut is made in the above strand, exposing 3 end and 5 end. See, at the 3 end, the nucleotides keep on adding, and as this moves down, it is releasing this DNA segment. See, moves down, it is releasing this DNA segment. Finally, this 5' end, it is released outside in the nucleus, and it forms a circular shape again. And once it forms a circular shape, it serves as a template, and it starts synthesizing a new strand. And this again, this is your new strand. The cycle. And once it completes the synthesis, the cycle may be repeated again. Again, it will go like this. It's called a rolling cycle. So the difference between the theta replication and the rolling cycle replication is the only thing. In theta, no cut is given to the strand. One. Number two, there is no origin of replication. And the replication, you can see, the synthesis starts only at one strand, right? However, in the previous one, the replication starts, the synthesis starts at both the strands. The rolling cycle, why it is called as this DNA. When the cut is made, it goes like this from 5 to 3 end. It makes a complete cycle and releases this. When it completes DNA, when it completes the synthesis, and it may undergo the same. Again, it will undergo the same thing. See, like this, and it will continue to form the DNA like this. That's why it's called a rolling cycle. This is released. The free segment is released and that 3 segment makes the circular shape and then carries out the synthesis. This type of replication is found in virus and other F1 elements in the bacterial organelles. Is that clear? And if you differentiate between the two, you will see a big difference. The difference is no replication bubble is formed here. The cut is given on one of the strands, and the replication, since it only occurs at one strand, you see, only at one strand. Other strand doesn't take part in the replication. However, in the theta replication, both the strands serve as a template, right? So this is your theta. So this is your rolling cycle replication. Now you have a linear replication. How does a replication take place in you and me? I said that in eukaryotes, we have multiple origin of replications, right? And in prokaryotes, we have only one origin of replication. Why you and me have multiple origins of replication? Because our cells contain large genome. Okay. See, yeah, here I have this diagram, which is a little bit ulta. The reason being, it is too large to have it in a portrait position. You see, this is your chromosome. This is your chromosome, right? Origin of replication. Bubble will form here, right? And when the bubble will form, this and this, these DNA strands are separated, and both will serve as a template. Both will serve as a template, right? See, both is synthesizing a new strand. That means both are serving as a template. Here also, and the synthesis continues, and finally you have a chromosome, 2 chromosomes. Is that clear? We have multiple origin of replication. See how many bubbles are there? 1, 2, and 3. And when the bubble forms, when the 2 strands unwind, each strand serves as a template for the synthesis of DNA. You see, this serves as a template and synthesizes this one. This one serves as a template and synthesizes this one. Is that clear? Do you understand this? Please unmute your microphone.
Yes, sir. Yes, sir.
Okay, you have to understand the template, because I said origin of here the replication has started. That means these two are tightly coiled over each other. They have to unwind first, uncoil first. When they are separated, yes, when they separate, they serve as a template. Both the strands serve as a template. Okay. So this is how linear replication takes place in linear chromosome. So I want to compare this theta replication, rolling cycle replication, and linear replication. But unfortunately, I cannot do it now. You may please unmute your microphone. We have 2 minutes, we'll have a discussion. Please unmute your microphone. I request all the students to unmute your microphone. Yes. Please say something. Are you getting this molecular biology, or it is all going over your head? Chinchole?
Yes, sir.
बेटा कुछ मॉलिक्यूलर बायोलॉजी समझता है कि या ऊपर सर के ऊपर से चला जा रहा है?
समझता है सर.
वैष्णवी
Yes, sir.
समझ रहा है क्या मॉलिक्यूलर बायोलॉजी? ये लेक्चर में मतलब डोंट एक्सपेक्ट कि लेक्चर में ही सब कुछ आपका क्लियर हो जाएगा. जो मैं वीडियो आपका फिर शेयर करता हूँ गूगल क्लासरूम में, प्लीज गो थ्रू दैट, प्लीज गो थ्रू दैट. और मैं कुछ स्टडी मटेरियल भी शेयर करूंगा. आफ्टर द एंड ऑफ द यूनिट आई विल शेयर द पीपीटी आल्सो. बट घर पे भी तुम लोग पढ़ाई कर लो. ये इट इज अ मॉलिक्यूलर बायोलॉजी. यू हैव टू हैव अ डिस्कशन विद मी एवरी टाइम. साक्षी, व्हाट इज योर टेक? व्हाट डू यू थिंक? आर यू पीपल सेटिस्फाइड विद दिस? कुछ समझ रहा है कि नहीं आ रहा है?
Yes, sir. I'm getting it, sir.
टेम्पलेट अगर तुमको समझ में आ गया, योगिता, जिंकल, लोहिता, गुंजन,
Yes, sir. समझ गया.
Yes, sir.
Yes, sir.
Template, you have to understand what is a template. So please, जब भी आपको कुछ लगे लेट मी नो. पता नहीं आज क्या दिक्कत थी. कल तो लेक्चर नहीं होगा, tomorrow is Saturday. But you people be ready for your presentation. I may have your presentation on next Saturday. Next Saturday तुम्हारा presentation. Let me go ahead. Next Saturday there is a presentation for you all guys. Okay, is that clear?
Yes, sir.
Yes. Drop your attendance here. You have to complete your CIA 3 on the next Saturday. We have 7 days enough to. और ये you don't have to just do your formality. जो भी मैंने पिछले CIA में कहा था, you have to follow that. Reference should be very proper. Nobody will do the paper reading as if it has to be student-teacher approach. You have to present very nicely. जैसे मैं पढ़ाता हूँ लेक्चर में, ऐसे ही तुमको पढ़ाना लेक्चर. You will be given a proper timing. मैं तो 1 घंटा दूंगा आपको, बेटा आप अपना लेक्चर कंप्लीट करो. तो कोई ऐसा ये फॉर्मेलिटी पूरा नहीं करेगा. ठीक है?
Okay, sir.